Okay, so we'll get started. Welcome everybody to the BNN webinar on sex and gender in biomedical research. We're really excited to have our speaker today, and we'll get started. I'm just going to give you a short overview of BNN, what we do, and then hand it over. So it's 2023, and we're at a time of unprecedented innovations, but with these innovations come also potential hazards to human health and the environment. What we do at BioNanoNet, or BNN, is to advise companies and research organizations to make their innovations sustainable and causing as little harm as possible to human health and the environment. So how do we do that? We're an association of nearly 70 members from Austria, but also the rest of Europe. And we cover the medical community, such as MedUni Graz, where we have a lot of participants today, to academic research, SMEs, and industry. At the same time, we're a company that offers services, and we do that largely through our EU project work. We work with our partners to design and develop innovative materials, products, or processes together, using safety and sustainability as our guiding concepts. To do so, we also provide them with the right connections or partnerships and alliances during the different stages of development and innovation until reaching the market. At the same time, we communicate with stakeholders and civil society to let them know how these achievements impact their lives. So what is this relationship to gender and biomedical research? So you'll see here that our work covers many different areas and topics, including human health. Take, for example, nanomedicine, from the development of new medical devices to in vitro diagnostic devices, to more target-specific treatments based on nanopharmaceuticals. We have projects like Phoenix OITB, which have created a one-stop shop for nanopharmaceutical development, or next generation microfluidics, which is enabling the development of microfluidic devices, such as for diagnostics and even for improving in vitro fertilization methods. Here and in many other examples, sex and gender can impact research results. So for our project partners and our association, we wanted to connect our community to an expert on this topic. That's where we turn to Professor Sabine Oertelt-Prigione. She is chair of gender and primary and transmural care at Radboud University, and she's also professor of sex and gender sensitive medicine at Bielefeld. In addition, she chairs the European Commission's expert group on gender and COVID-19, and was previously a member of their expert group on gendered innovations, which is a topic that I think will resonate with a lot of you. So without further ado, I'll hand over the mic.
All right, well, thank you very much for the invitation and the opportunity to talk to you today. I really appreciate your interest in the topic, and I do think that it connects with a lot of the initiatives that you just mentioned on so many different levels in the biomedical continuum, and I hope that everybody can take some message home today. And as we said before, if there are any comprehension questions during my talk, please feel free to put them in the chat and we'll address them immediately. If there's anything bigger and broader you'd like to discuss, we'll keep it for the discussion afterwards. If after my talk you still have some questions or ideas that come up later on, I'll also leave my contact details at the end of the talk, and feel free to get in touch with us if anything comes up later. So what will I talk about today? I'll address quite broadly what sex and gender mean in research and what their impact could be. So I'll start with what the gender dimension is from a European standpoint. That's the definition that's being used by EU Commission. I will then move on to defining what actually sex-specific analysis in research and innovation entails, what that means, and what that means methodologically. We will then move on to gender-specific analysis in research and innovation. These are two distinct things that can interact, but if you want to analyze it, you might have to go two different routes. I'll show you a few examples of what can possibly go wrong if you don't do it, hoping to convince the ones that might not be convinced yet. And then, of course, a last thought about the connection between, well, structures in research and the content we're producing. All right, this is a short historical path to where we are right now. If you look a bit at the history of sex and gender sensitive medicine, and I'm not going to walk you through this whole thing, it's just to show you how much it has grown over time. It really started in the 60s and 70s with the women's health movement, starting pretty much everywhere in Europe and in other areas of the Western world. And from here, moving on all over the globe, and also slowly moving, especially in Northern America, into structures and into structural changes. It took us much longer to implement comparable things in Europe as well. But you see here the NIH already getting active in the 80s and 90s by establishing also organizational pins that could support especially women's health at that point still. And then the transition into the 90s and early 2000s, and we see that the European Commission is picking the topic up more and more. And what is interesting is at European level, it was not so much a focus on women's health from the beginning, but actually a broader focus on sex and gender in general. And so also a more holistic approach to the topic right from the beginning. You see here a number of relevant milestones. What you see here in these last years is especially more and more guidelines coming up, more and more structural support coming up, websites, gendered innovations has already been mentioned as well. We have several resources that are available to all of you, and you see how this has been growing and is still growing over time. And I hope that today we'll put a little pin in for you as well to add some more information. Now, why do we need a focus on sex and gender at all? And this is not what I'm gonna tell you. Of course, I agree with what they say, but it is really about what the funding organizations are telling you. And what you see here is what the European Commission in their research framework says, what the Canadian Institutes of Health Research say, and what the US NIH say about why they would like you to incorporate sex and gender into your work. And you see it is about the improvement of scientific quality and societal relevance. It's about improving rigor, reproducibility, and generalizability. And again, here, reproducibility and rigor and transparency. So they don't take it lightly. We're not talking about some minor thing that you can do on the side. It is really big topics in research that can be addressed by focusing on sex and gender. And one important aspect is reproducibility, and we'll get to that again later. The other one is excellence in trying to produce the best research, and of course societal value in trying to produce research which is relevant for everybody and not just for a minor group within society, but actually for as many people as possible. What you see here is how the European Commission goes about it, and I like to point out that it's not just about integrating sex and gender analysis in research and innovation, which is what we're going to be talking about today mostly, but there's also two additional layers that are being mentioned here. So one is fostering the gender balance in research teams. So this was explicitly said in Horizon 2020, but it's carried over into Horizon Europe. And then we have ensuring gender balance in decision making. So we would like to see equity here as well. And what the EU Commission is showing you here is that you really have two layers to take into account. So on the one hand side, you have context factors, who gets to do research and who gets to make the decision about what is being researched. And then you have questions about content. So how is it being done? And as I said before, today we're going to focus mostly on that, but we cannot ignore this aspect because it also impacts what happens in here. So let's focus on the content for today. Now, when it comes to sex and gender, I think it's important to clearly define the terminology at the beginning already, because for many years and still today, we just did a literature analysis which showed that even up to today, these terms are still conflated, which does not help the science and also does not help the societal discourse about this. So we talk about two different layers, I would say, that are intertwined, as you see here, but still address slightly different aspects. On the one hand side, we have sex, which are mostly biological factors, and it's the first part we're going to be diving into today. based on genetics, genitals, and gonads and hormones. And then you have all these gender aspects, and these are only a few that interact and are intertwined with sex. You have on the one hand side gender identity, which is what the media mostly talks about, which means, are you a woman, are you a man, are you a non-binary person? Then we have gender norms and gender roles, which is really what society expects you to be in terms of masculinity and femininity. How have we defined gender in our societies, and what does that mean, and which kind of assumptions do we associate with that? And then we have gender relations, which is really the interaction between the two, between your identity and the norms and the expectations that you're facing, and how does that affect interaction between humans. And then, of course, there's other dimensions that I'm not going into detail today, which is gender expression, for example. How do you express your gender identity? We have institutionalized gender. How is gender intertwined with the way our organizations work, our laws work, and so forth? But that's just to say that we have many different things we're looking at, and we will now have a closer look at what that means. The first point to think about when considering sex, and this is what we're going to be talking about for the next few minutes, is that every cell has a sex. So every cell has sex chromosome, and this is what is meant here, but cells are also exposed to hormones throughout their life cycle. And even when you take them out of the body that has harbored them up until that point, even if you put them in culture, they will still keep some of these characteristics. They will keep definitely the genetic characteristics, they will keep the epigenetic characteristics up to a certain degree, and most likely they will keep the influences of the hormones they have been swimming in up until that moment. So this is something to keep in mind, that even if you work with cell cultures, you still see that. Currently, when we operationalize sex, we use 3 levels, the 3 Gs they have been defined, because we look at genetics, we look at hormones and gonads, that's why the second G, and genitalia. And these are 3 ways to operationalize sex. The first one, if you want, is in, if I can put that in quotation marks, the simplest or the most binary still of all 3 of them. So we assume in biomedical research that pretty much most of the world will fall into 2 categories. You're either XX or you're XY, which is true to most degrees, although there are exceptions, and there is a certain degree of the population which will have either an XY, an X0 correlate, so you have only 1 X chromosome, or has multiple copies of the X chromosome in their cells. So it can be XXY or XXXY. These are relatively rare. So it is true if we assume that most of the population will be born with either an XX or an XY correlate. However, our bodies are dynamic and they change over time. And one of the points to keep in mind is that especially the cells in our body that reproduce a lot or that get exchanged a lot, for example, blood cells or fibroblasts, do feel when they get older, and some of these processes don't work as well as our bodies age. And what I mean by that is, for example, if you look at the circulating blood cells in people above 60 or 65, there's quite a number of these blood cells that will only have an X chromosome, for example, or there might even have a second copy, but usually they lose one of their chromosomes. A Y monosomy doesn't resist very long, but X monosomy has been described quite frequently and has also been associated potentially with cancer or autoimmune diseases. So what that means is that even if our bodies start off as XX or XY, it might well be that over the course of time, some of our cells will end up being X0. So even a paradigm of XX, XY is not that fixed in time and can change. Of course, you will only find that out if you actually look for the sex chromosomes in your cells. The second layer to look at is the hormones or gonads. So you can define sex by looking at the hormones that are expressed in the body of the person you're investigating. It is important here to consider that hormones change. They change throughout the day. So if you measure testosterone in the morning or in the evening, it will have very different concentrations. They change throughout the month And the easiest example here is the menstrual cycle. And they change throughout our lives. As we get older, we produce less hormones, and that goes for all of us. And also the balance between these hormones might change. So all individuals produce all hormones. We, apart from some exceptions, of course, that there are cases in which hormones are not produced or you are resistant to hormones, but those are more of an exception. In general, all bodies produce all hormones, but we produce them in different concentrations. So when you decide to define sex based on hormones, you have to make choices and decide which kind of hormones you're going to measure. So are you going to measure the most bioactive ones? Are you going to measure the most abundant ones? And where are you going to set cutoffs to define male and female participants, for example? Because as I said before, these things potentially can vary. But the fact that they vary doesn't make them less important, because, for example, even if you're not studying the menstrual cycle, it might be important to know if a person is in one or the other phase of the menstrual cycle, because being in either the follicular phase or the luteal phase might have an effect on the T-cell response of that person. So while you might be studying response to viral pathogens and have no interest whatsoever in menstruation, it might still be important to know whether that person is cycling and in which phase of the cycle they are, because this can have an impact on the T-cell responses you're actually measuring. Because in the follicular phase, you will see a preponderance of Th2 response, and in the luteal phase, you will see a preponderance of the Th1 response. So if you only analyze the cells without considering that, you might actually miss relevant information. The third level, and that's probably how most of us have been attributed to one sex or the other, is we're born as babies. Somebody looks at us, checks that we have 10 fingers and 10 toes, looks at our genital and tells us, okay, you're female or male. Now, in most cases, that works pretty well and has worked pretty well for centuries. However, there are individuals that cannot be clearly attributed based on their apparent genitalia. So these are individuals that today we call exhibiting differences in sex development, DSD, or intersex, as they frequently want to be called themselves as well. The prevalence of intersex or differences in sex development is quite different depending on which statistics you look into, whether they are medically assessed or self-reported. So as you can see, we have percentages that go from 0.07 to almost 2%. There are a number of conditions can be the medical basis for DSD, and you see some of those here. I put a mole here, not because moles are very cute animals, they are, but because the mole is a very interesting example of a mammal that is always intersex. So the female mole is born intersexual. And what that means is moles live under the earth. They need very strong muscles to be able to live under the earth and have their mole lifestyle. And to maintain their mole lifestyle, what nature has done with them is that female moles who are perfectly fertile have testicular tissue in their womb. And this testicular tissue actually produces testosterone. And this testosterone impacts the strength of their arm muscles, which allows them to live their mole lifestyle. So what that shows is that there are other examples in the mammal world of, well, animals in this case, that are by constitution intersexual. And we don't even know how many there are because the moles have been discovered actually by accident some years ago. It was not what the researchers set out to look out first. And this was only found out a few years ago. So we might find a lot more animals that have these characteristics and potentially might even find more diversity in humans as well if we keep looking and investigate this somewhat further. Now, that's just to say that measuring sex is not as easy as it seems. And the choices you make in terms of what you're measuring might potentially impact your results. But what does that mean for example, for cell cultures? So how can that affect the work you do in the lab? Well, first of all, you don't need to answer to this, but how many of you do actually know the sex of your cell cultures? As I said before, monosomy can appear throughout our lives. We can have circulating monosomal cells, but we will only find out if we actually look for them, if we actually measure the karyotype of these cells. Furthermore, karyotypes tend to change, especially if you keep cells in culture for a long time. And this has been described mostly in the oncological field. We have some tumor cells which we've worked with for decades that have been demonstrated to have quite interesting and quite funky karyotypes after many, many passages. So they can have multiple copies of autosomes, they have multiple copies of sex chromosomes. So these cells in culture change their karyotype, which can of course affect the way they react and can affect our results. If we, for example, reprogram cells and we make organoids, this is a process that also can change epigenetics and specifically X inactivation patterns, because what we do through the reprogramming process is we reactivate the X chromosome and then inactivate it again, which can have an impact on the methylation patterns. The type of media we're using can impact our cell cultures, and of course, depending on where the cells are coming from, the methylation might be different. What we still have questions about is, for example, which kind of effect different hormones can have. So whether they're synthetic or whether they're purified. In culture, does that affect the way our cells grow? Does it affect our experiments? And of course, also there could be intrinsic differences based on the media you're using. So all of this can affect the experiments. In addition to that, also our animal models that we like to work with are not completely unaffected by this. So for example, depending on how many animals you have in a cage, they will interact with each other. And this is different depending on whether they're female or male animals. They have social dynamics. The diet we are giving them, are they getting soy-based chow? So soy is a natural phytoestrogen which has estrogenic effects. How do temperature and sound and lighting affect, for example, their circadian rhythms? I said initially that testosterone is different in the evening and in the morning, and that has to do with the circadian rhythm, which on the other hand is connected to, for example, light and temperature. And of course, there's the big question about the researcher. Do researchers actually impact the animals they're handling? And while in the past, everybody would have said, well, this is kind of a ludicrous assumption, turns out it's not. Because, and I would really recommend that you have a look at the seminal paper about this phenomenon, which was published in 2014 out of Jeff Mogul's lab in Canada. Apparently, the examiner sex has an impact on the examined animal as well. Because what they saw in this group, which studies pain in animals, was that depending on whether the examiner was female or male, the animals interacted differently and showed pain in a different way. So basically, the assumption here is that the animals smell pheromones in a cross-species fashion, and this was actually the big discovery here. And depending on the pheromones they're smelling, they show pain differently, depending on whether the examiner is female or male. Of course, this has an impact if you're studying pain, because you would like the animals ideally to show you in the same way if they're feeling pain or not. And if this changes depending on who's doing the experiment, you might have a problem. This has been replicated by now in several other groups independently of this first finding, and is especially important if you are analyzing animal behavior. So when you are looking at the animal interaction with each other or with the examiner and you're examining behavioral changes, then this might be an important factor to keep in mind. So when it comes to reporting of sex, and this is the conclusion of this first block, there are a few things that you should report. So first of all, the rationale of the study. What are the potential effects of sex on the performance of your experiments? What could it do? You should report your experimental setup. We would like to know the total number of subjects, of cells, of animals broken down by sex. That should be your table 1, and ideally you would like to see that everywhere. There are additional confounders that you can describe, which most of the time are not really described. I mean, we know a bit about how animals are fed, but not a lot about the housing conditions, the light and dark, the temperature. It's a question mark if in the future we will have to know about experimenter data. We would urge you to analyze your experiments by sex, and ideally having also some interactions if they're meaningful, for example, sex and age, or I don't know, sex and ability or disability if you're doing social studies, for example. And then this is a big one, and I say that, and it should be a given, and it should be very simple, but unfortunately this is still not happening to the degrees we would like to see. Report your analysis in a disaggregated manner, and if you only put them in the supplementary files, we'll be happy with that. But please report your data in a disaggregated manner, and of course discuss the implications of what you found. Now, this was analyzing sex, and as you've seen, it's already more complicated than it might seem at a first sight. Let's have a look at gender, and I'm only gonna touch upon some of these aspects because the analysis of gender in the biomedical field is still something that is developing. And we're still working on the methods, and it is a field that's quite exciting at the moment because many things are happening, and then we're developing many new methodologies. Now, it is already tricky to start measuring gender identity because for the longest time in the questionnaires we've given our patients, the questions we were asking were simply, are you female or male? Which of course could be interpreted as your biological sex, but could potentially also address your gender identity. Now, in most cases, the two will match, but there are a growing number of cases where they don't. And since we never asked in the past, it is also a bit more difficult to have a good baseline to start with. So several statutory surveys in different countries are picking up the two-steps approach question, where you will be asked what sex you were assigned at birth, so female, male, or intersex, what we just talked about, and what your current gender identity is. A woman, a man, you're non-binary, you're genderqueer. Ideally, this gender identity question should be piloted first with the population you're actually using your questionnaire with, because the expression of gender identity and definitions of gender identity might vary depending on where you're asking these questions. And there might be words or definitions that are used in certain communities and not used in others. So this is the first one, to even give you a glimpse of sex and gender. Does this matter? Well, there's even questions where both of these might matter. So for example, when I investigate pharmacological side effects, I might need both of these questions. Because if I want to know if there are biological differences in the likelihood of developing a side effect based on, I don't know, the metabolism of your liver, I would like to know about biological sex, because most likely it's a biological interaction. If I'm actually collecting data on the reporting patterns of a certain side effect in primary care, I might be more interested in gender aspects, because we know that, for example, reporting symptoms might differ based on gender, and also the likelihood of being taken seriously with your symptoms might differ based on gender. So for the same question, pharmaceutical side effects, I might actually need both these questions to properly address what I'm looking for. Then the next layer is gender norms or gender roles. And there's quite a number of questionnaires that have been developed in the 70s and 80s about this. It is not really something that is very active right now, but some of these questionnaires are still being used today. And what I'm showing you is the BEM sexual inventory, which, although it has been developed in the 70s in Berkeley, is still being used today. And we have even used it in my group still to see in how far this could be helpful or not towards the work we're doing. The way it was developed was that in the 70s, psychology students in Berkeley were asked to define typical masculine, feminine, and neutral items. And based out of that, Sandra Bem developed these questionnaires which allow you to classify your participant population into 4 groups. So the sex typical female, sex typical male, the androgynous, and the non-defined, the neutral group. All of these have different characteristics. They do correlate in one way or another with health-related quality of life. Of course, we can have discussions about how relevant they could still be today, but most importantly, how relevant they are in certain cultures in certain times, and which way they can be adopted or not adopted in different parts of the world. Regarding gender relations, you have a different facet again, where you can look at how people interact. So gender relations are frequently measured in the private space. You can also measure them in the professional or in the public space. And what we're looking at is really how does my self-perception interact with the perception that others have of femininity or masculinity, and how far does that change our interactions and our behaviors. So as I said before, a lot of times what is investigated is, for example, gender relations in the private sphere. So how are, for example, how are tasks in the home attributed to different partners. So this study was done in Sweden in heterosexual couples who were asked, how equitable is your partnership? So how equitable is the division of tasks in your partnership? And it shows you something quite interesting. You have the self-perceived, which either are the brown bars, which is what people say. So if you ask them directly, well, how equitable is your partnership? You have the men that will tell you mostly quite equal or totally equal, and some will tell you not equal. And you have the women that say, well, it's quite equal. Some say totally equal, a bit less than the men, and some say not equal, some more than the men. But then if you use actually a somewhat more objective index where you ask them specific questions like, how many hours do you spend on doing housework? Who is in charge of that housework? Who stays at home if your child is sick? How many paid work hours do you do in a week? And so forth. And construct an index from that and compare it to the self-perceived, you see that there's very interesting differences. First of all, the quite equal decreases in all of them. Interestingly, in the women that said less totally equal, actually there was a higher number of objectively totally equal partnership, but also objectively not equal partnerships. In the men, the bar with the not equal increased the most, and these others decreased. So what's that to say? Well, if we are just asked if our partnership is equitable, or if our, for example, professional gender relations are equitable, we will probably give slightly different answers than what we can measure. And that has to do a lot with social desirability bias. So as I said, the study has been done in Sweden, where gender equity is a societal value. So it's something you strive for, and you strive for your partnership to be equitable. So of course, you're more inclined to say, yeah, of course it's equitable. But then if you measure it, it might look slightly different. So that's just to say that if you want to look into equity in relations or in gender relations in general, it's always quite helpful to ask a direct question, but also have some kind of measurement tool that will give you a different information. Now, how have people gone about bringing these things together? What I'm showing you here is one of the attempts in around 2013, 14, 15 to come up with gender indices. And this was done by Luis Pilota's group in Canada and has definitely started a whole new and spiked interest into developing gender indices, which are questionnaires that can bring together different gender dimensions. And what you see here is what they in fact asked are, for example, gender relations, as I just said, the primary earner in a household, the personal income, the number of hours spent doing housework. You see the gender norms here, the gender, the BEM sex role inventory, and so forth. But you also have factors like level of sex stress at home. Now, what this index will spit out is the probability between 0 and 100% for each patient to be a woman. Which is a bit problematic, especially because since the times of Sandra Bem, what we were actually doing was disentangling this and showing that there's at least that everybody can have femininity and masculinity. But it is a first attempt, definitely, to try to bring together different gender roles. Of course, people have also started questioning if these are the right things we're looking for. And for example, Matthias Nielsen and Londa Schiebinger in Stanford have developed a completely different measure for gender, which takes into account different measures. You have, again, the work strain or the caregiver strain, but you also have other aspects like emotional intelligence, social support, and discrimination that come up here. And what you see here is that this construct, for example, is again able to discern between men, women, and non-binary individuals. The applicability of all these questionnaires in health is still to be investigated. And as you see, most of them are quite recent, and it is a new trend that has started in looking into different ways of measuring gender, which we will continue see evolving. Now, I talked about sex and gender separately because I wanted to show you why it is important to measure them and how you can potentially measure them, because I assume that not all of you will be interested in measuring both, and not all of you will have to measure both. But if you do measure the one or the other, you should know what you're doing and you should be able to take them apart quite well. So I think that's the important message here. You should be aware of what you want to measure and pick the right instruments for what you want to do. But what I want to say to conclude this first block before we move on to the examples is that, of course, sex and gender are intertwined in your daily life, right? We have in the beginning, this is us as babies, we're born with all our sex features, genes, genitalia, and hormones, but then we grow up in a gendered system, in our family, in our community, in the institutions we have to deal with, and the structures and policies. And what we see is all of these are gendered, which means we can basically not grow up without being influenced by gender. And this again impacts our society overall. We know that in our societies, whether we want it or not, we build certain hierarchies where gender also interacts with other categories like ability or age, race, and class, which lead to the fact that access to healthcare is different for different people. That we have differences in exposure to risk factors, we have differences in health behaviors, we have differences in access to the healthcare system, and we have differences in how data is collected and which data is collected. So this happens at the aggregated level, but at the end of the day, it impacts the health and the sickness of the single person. So what can possibly go wrong if you don't do it? I already showed you this one, so I'm only picking it up again because I want you to look at it from a slightly different angle in terms of reproducibility. So when I described that the investigator can have an impact on the investigated subject, this is of course quite striking and quite, well, scary maybe for some who work with animals and who look at their behavior, but it might be a question of reproducibility. It might be a question of what do we need to report in order for others to actually reproduce the experiments the way we have done them. So the fact that even an experimenter can impact and their sex can impact the way animals behave might be something we will have to think about in the future, or at least in your specific lab, in your specific environment, if you see something that doesn't make sense. This is probably the most well-known and the most common aspect that is being discussed. If you ignore sex and gender differences, you have a quite high risk of delayed diagnosis in clinics. And there are some examples that are well-known. One very well-known one is bladder cancer. So we know that a lot of women experience a delayed diagnosis of bladder cancer because if they go to their GP, they go to their doctor and say that they have seen blood in their urine, what we first think about is a urinary tract infection, a cystitis, which is epidemiologically speaking also the most likely explanation. However, if after 1 or 2 cycles of antibiotics and still blood and urine, nothing happens, it might be worthwhile to pivot and start looking for different explanations and not wait some more months while the patient actually has bladder cancer instead of a UTI as we initially thought. When it comes to very common diseases, like for example, asthma, although women, especially in older age, refer more symptoms, they're less likely to get diagnosed. In young years, it is often so that the typical wheezing, which is the typical symptoms we look out for when you have asthma, is much more common in boys than in girls. And so this can lead to an underdiagnosis because in young girls, a lot of times asthma presents as a dry cough at night and not necessarily as wheezing. When it comes to other diseases, like for example, osteoporosis or autoimmune diseases, we associate them with women, with being female, with a typically female patient, which leads to under or delayed diagnosis of men. So 30 to 40% of men over 70 have osteoporosis, and yet we don't screen them regularly. And we most of the time only find out if they break their bones, rather than finding out earlier and give them supplements that might prevent some of those fractures. Same goes for autoimmune diseases. Autoimmune diseases are more common in female patients. Most of them, there's a few exceptions, but most of them are more common, which also means that more frequently the diagnosis is delayed in male patients because we don't think about autoimmune diseases at first. And then I really hope that I could take this kind of vignette out of my slides at a certain point. It's an Israeli study from several years ago where they asked patients with multiple sclerosis, which is notoriously difficult to diagnose, which kind of specialists they have seen in their journey before actually being diagnosed with multiple sclerosis. And most of the male patients will tell you that they were sent to an orthopedic surgeon at a certain point, while the women will tell you they were sent to a psychiatrist. And I really hoped I could take this out, but then COVID happened and long COVID started to happen. And we saw a lot of this all over again. And so, yeah, maybe I cannot take it out yet, but maybe in the next generation. Now, of course, we're moving into the digital age and everything will be done automatized by computers, which will use marvelous AI and will, through machine learning processes, solve all of our questions. Well, we are in this world to a certain degree already, and we have seen a proliferation of apps and chatbots in the biomedical field that has been unprecedented. Unfortunately, only some of them are actually good, and a lot of them have raised questions, to put it diplomatically. Exactly like this one, which, however, is a brilliant example of why it's important to look at sex and gender. Now, this chatbot was developed to give you an indication of which kind of symptoms, what kind of disease could be associated with the symptoms you're experiencing. And so if you told the chatbot that you're a 56-year-old, that you smoke 10 cigarettes a day, you have a painful left arm for the last hour, and now you're feeling sweaty and sick, and you tell it that you're a man, the answers you're going to get is, well, you could have pericarditis, which is an urgency, and you should go to the emergency department immediately. The second one is you could have unstable angina. And again, you should go to the emergency room. Number 3 is you could have a gastritis, a stomach inflammation. You can go to your GP tomorrow. 4th one is you have a panic attack, stay at home, breathe. And number 5 is you could have a heart attack. This is also something that is treated in the emergency department. Now, after reading this, probably you're already on your way to the emergency department if you are a man. However, if you told the bot that you're a woman, the first 2 answers you get is 1, you have a panic attack, breathe. And number 2, you might have depression, which you will probably have if you didn't know you had a heart attack and didn't go to the hospital in time. Now, the chatbot is not trying to kill you actively, or at least I assume it's not. You can, if you want, look up this hashtag on I mean, I don't know in how far Twitter still is able to do that, but it was possible some time ago to actually use this hashtag and find some other brilliant examples of what this chatbot spit out. But this is just to say that it's great to have huge amounts of data, and it's great to mine them to help us make clinical decisions or make predictions, but we should also know which kind of data we're using. So as I said in the slide before, the delayed diagnosis due to assumptions about who has a disease and who doesn't have been problematic in the past. So if we use this kind of data to train our algorithm and our AI, of course, what it does is it will reproduce what has been done historically because it learns from historical data. So this doesn't mean that historical data is intrinsically bad. All data is biased in one way or another, and assuming that data is not biased is just, I would say, somewhat foolish. The point is, what do we do with these biases? So how can we incorporate attention to the existent bias, and how can we use that productively in order to avoid reproducing the biases we already have in our historical data? Interestingly enough, this doesn't just go for sex and gender, it also goes for other intersectional factors. We have had quite interesting report about pulse oximeters that didn't report oxygen saturation appropriately, and this was of course especially important during the pandemic. And that went for both potential differences between men and women, but in addition to that, potential differences due to skin color. Because a lot of these oximeters actually read oxygenated blood, and they don't work as well if skin is darker. So if you don't factor that in, it might be a problem to have your devices actually work appropriately, and not just for women and men, but actually for different women and men of different ethnic origin, with different skin color, and possibly other differences. Now, if I haven't convinced you by now that it should be relevant and you should look at sex differences, I have one example, which is another historical example that could possibly convince you. So this is data from the 90s. It's the DIG study about the effectiveness of digitalis in treating heart failure. And what the question here was at the time was, first of all, in the safety study, in how far is digoxin, which is the name of the drug, in how far is digoxin as safe as placebo? And when you look at that, you'd say, well, pretty good. People using digoxin have the same mortality for any cause than people using placebo. So this makes the drug quite safe. You don't want a drug that kills people more than a placebo would. You actually want a drug that helps people and doesn't lead to their death for other causes. It's a pretty big study. You see almost 7,000 participants. So people were quite confident that this is a quite safe drug and we should be using it to treat heart failure. So they started using it to treat heart failure, which worked quite well for men, and saw that especially women started getting a lot of side effects, and sometimes severe side effects. So what they did was 5 years later, they did a post hoc analysis of the same data. They again took these almost 7,000 participants, but this time they split the data in male and female patients. And what you can see here is that the male patients do indeed much better with digoxin than with placebo. So their survival is actually better on digoxin than on placebo. But if you look at the women, their survival is actually better on placebo than on digoxin. So this was not exactly ideal. But why did we not see that before? Because the women were only 25% of the participants. So this larger difference was only seen in 25%. And the smaller advantage was seen in 75% of the participants. So if you lump them all together without splitting it up, you get this. You get a curve where you don't distinguish anymore. Now, is digoxin intrinsically bad for women? No, it's not. It's definitely not. And it actually works quite well in women as well. But digoxin has a relatively narrow window of effectiveness where it works well and it doesn't give you side effects. So what you need to do is you need to monitor and make sure that the digoxin concentration is in that window. And if you give everybody the same dosage, the likelihood of overdosing women compared to men is quite high. So you need to monitor that in order to make sure that everybody gets the right concentration. But of course, if we hadn't done the separate analysis, we would have never found out. This wasn't just digoxin. This is a slide with 10 drugs taken from the market in the United States at the end of the 90s, beginning of the 2000s. And in 8 of these drugs, the incidence of side effects was much higher, and in part deadly side effects in women compared to men, mostly because in preclinical studies, female animals were not considered. And then up until the beginning of the 90s, women were excluded from clinical trials. And hence, we had no information about the effect of some of these drugs in women before they reached the market. And then once they reached the market, they were, however, ever handed out to everybody. So unfortunately, the only way to find out about these side effects was actually pharmacovigilance after the drugs had been released, because we had no information about their effectiveness and side effects in women prior to release. Now, this is not the same anymore today. Since the early 90s, women are again included in clinical trials, and their numbers have been increasing ever since. So this is quite different today. But still, not everything is perfect. Because then, for example, when things like a pandemic hit and we have to make fast decisions, we tend to sometimes fall back onto our defaults. And what we saw, for example, was what happened with COVID-19 trials. So together with colleagues in Denmark, we actually looked at COVID-19 trials registered in clinicaltrials.gov, which is a large database where pretty much all large multinational clinical trials are recorded. We looked at all of those that had been registered between January 2020 and January 2021, and those were more than 4,000. And surprise, surprise, only about 1 in 5 actually reported sex and gender as a recruitment criterion, and only 1 in 20 reported that they would consider sex and gender upon analysis. Now, one could argue that when you register your trial, you don't put all the information out there. So maybe the analysis plan is not as specific as it will be, there's information missing and so forth. Yeah, that is true in part, because we also looked at the published trials in January 2021. So most of these registered trials might never see the end. So they have been registered because somebody wanted to do the trial, but then for one reason or another, either you don't manage to recruit enough people, or it turns out that your hypothesis has been scooped by somebody else, or for one reason or another, many of these registered trials never saw the light of day or were never concluded. But some are, and then get published, and this is what we looked into. So we looked into the pharmacological trials in January 21, and yes, the situation is better than the 4%, but it's only marginally better, because we go from 1 every 20 studies planning to look at sex and gender upon analysis to 1 in 5 studies actually doing it. So, which is better, but it still means that in about 80% of these publications, we had no information about potential differences in effectiveness and side effect between female and male participants. And sadly enough, as you can see here, this did not change throughout the pandemic. So it's not like we had information about potential effect of sex of COVID-19, which, by the way, we heard a lot about in the media, and so people did it more. The patterns were exactly the same throughout the year. Now, how do you mitigate that? So what would I like you to do? And these are my concluding words. When it comes to this section, you should know which kind of samples you're working with. As I said before, ideally you should know the sex of your cells, you should know the sex and the gender identity of your participants, and so forth. So have a clear idea of what it is or who it is you're working with. I would argue you should include at least both sexes. In some cases, you might think about including intersectional aspects or including intersex individuals, but at least include both sexes. And think about how to conceptualize gender. So if you want to measure gender, I showed you a few ways of potentially doing that. You don't have to do all of them all the time, but you need to think about what it is you want to know and which kind of gender dimension might impact your results, and hence which one you want to measure. I would advise consulting an expert. There's quite a few people out there who work on these things, so go and reach out. As I've shown you, it's not as easy as it seems, because a lot of times people now think, okay, I have to do this. The granting agency is asking me to do it. We'll just compare females and males and we're done. Well, yeah, I wish it was that easy. It's not, but there are experts out there that can help you. You should disaggregate data, and if I haven't convinced you with a DIG study, I don't know what I could convince you about this. You should stratify your analysis by sex and gender, and please report it. I made this point before, but it is still puzzling to me how we can have table 1, which tells us who participated in the studies, and then we don't have disaggregated results about how the things studied affected our population. And of course, we would love you to spread the word like we're doing today. Now, 2 last thoughts before I leave you. I talked a lot about content today because this is what we were mostly trying to address, but I have 1 slide about context, because there's always a question in how far do these measures impact what we actually do. And this is a large study done several years ago on 1.5 million papers in PubMed, where Matthias Nielsen and colleagues looked at in how far does the gender of, and specifically being a woman, of the authors affect the likelihood of having a sex and gender sensitive analysis in the paper. And as you can see here, it's quite clear. If there were women as first authors, last authors, or members of a group, the likelihood of having a sex and gender sensitive analysis is much higher than if they're not present. So what that shows us is that we're all different as researchers. Of course, we try to be objective, but the notion of total objectivity is, of course, naive. And we all ask different questions based on who we are and which kind of things we deem important for ourselves and for our patients, and also the things that we see that don't work and we would like to change. So having diversity at the table, and that doesn't just go for having more women at the table, it actually goes for having more diversity on many different levels, will provide us in the end with a more diverse result that could help us achieving that societal impact that the funding agencies were asking for. This is being addressed more and more at the European level. As you might know, if you're trying to apply for grants, you know that at the European level, we have a novelty this year that now you need gender equity plans in your organizations to actually be eligible for European funding. We already had the integration of the gender dimension, which is content, what we talked about today, for the award, and gender balance as a ranking criterion. And interestingly enough, in these gender equity plans, there's 5 aspects you should consider. And they pulled kind of a bit of a hidden thing here, because you see on number 4, again, we have all these structural aspects, and number 4, then we have integrating the gender dimension into research and teaching. So in your gender equality plan, you might also have to address the content part. So as I told you, the structural part and the content part are intertwined. As I can show you here, the context and the content are not completely separate. And this is something to maybe also keep in mind in designing your project. And with this, I thank you very much for your attention, and I'm happy to take any questions that you might have. Thank you.
Great, thank you so much, Professor Oertelt-Prigione. We had here in our office some literal gasping and nervous, shocked laughter at some of the results that you showed us. So we would love to hear some questions from the audience. You're welcome to write it in the question function you can see there, and we can read them out. We can also unmute you so you can ask yourself, but we can go to that in a second. I'll start off with a question because I'm not a researcher myself. So I'm wondering how, when you're actually doing your study, how difficult is it to disaggregate the data? Is it that it really in the end takes so much more time than not doing it? Or is it just that people are not aware of the importance and it's just kind of one extra thing to do?
It actually doesn't take that long, and it's, I will put it like this, there's a few layers to this. I think if you want to do a proper sex and gender disaggregated analysis, you need to consider it from the planning process, especially if you want to measure gender, because I think when it comes to sex, a lot of times we don't know how it is defined, and I showed some things about definitions, but in most cases we have some kind of information about sex of the participants. So actually doing a disaggregated analysis is really not that much additional work, and it is data that's there. So in a way, it is relatively easy to do. It might be underpowered at times, depending on how you designed your study. So that's why it is important to keep this in mind in designing your studies, because it could affect the ability to do it if you haven't considered it in the beginning. If you want to measure gender, on the other hand, it's more tricky. It's more tricky because in our standardized data, we usually don't really have much information about gender. There are some proxies you can use, as you saw, for example, the time people spend on working, or the time they spend on, I don't know, sometimes there's questions about childcare and some questionnaires, especially the large sociological ones. But still, these are all kind of proxies that you're using. So if you actually want to do it properly, you might have to add new questions. So measuring gender is more complex. But when it comes to a sex disaggregated analysis, especially if you have a large population, you should have enough numbers to have a robust analysis, and then it's really not that complicated. What happens at times is researchers might be doing it and they don't find any differences, which is perfectly fine. You don't have to find differences all the time. There are no differences all the time. Sometimes there simply aren't. But it's still not reported, and I think that's also an important aspect. If you did the analysis and you didn't find any differences, let us know, because this is also part of the research process, is also knowing, okay, people have done this, there was no difference in the study. But if it is not discussed and if it's not addressed, I will never know. And this kind of takes us back, and especially with the consideration of, sparing use of data and kind of trying to reuse data that's available and not collecting data that is unnecessary. With all these considerations that we're having about data in general, the better we make this data accessible, and should be one of the essential FAIR principles. Unfortunately, it's not always executed well. One of the essentials should be to have this information so that sometimes you don't have to repeat analysis maybe.
Yeah, thank you so much. We have a question here from Joanne. I will unmute you. Can I unmute? To see if you want to ask it yourself. Or Simona, are you able to unmute her? Okay, otherwise I'll just read out her question. She says, hi Sabine, how can researchers work to influence their universities in addressing these problems with including sex and gender dimensions in research? I've worked in a number of universities and it is not being addressed on a ground level.
Well, I think you see, I have to briefly plug in my computer, otherwise you're gonna lose me. So
Don't go yet.
I'm gonna answer your question. So I think what helps right now is, of course, that funding agencies are asking you to do that more and more, and universities are more and more dependent on third-party funding. Whether we're not going to discuss what we think about that today, but in principle, third-party funding is becoming more and more influential and essential for universities. So I think that's actually an angle that they might pick up on, because it is something that has to be done. And while in the past we have seen very nice and common formulations of people thinking, okay, this is boring, I just write in, we will include an appropriate number of female and male subjects. I don't know how many grants I've seen with that formulation. It's not good enough anymore, especially if you end up in a competitive arena like the EU, but also more and more national agencies are seeing more competition, and more national agencies are actually starting to pick this up. It might cost you points, and you cannot, well, yeah, you don't get the grant, but you cannot really accept that aspect anymore. And I think that's one of the things that works relatively well with universities, is making clear that the researchers need to be informed about this, because this is something that becomes essential for them in getting the funding they need, which the universities themselves cannot provide a lot of times. And goes also for research institutions. Another aspect is that there is a growing number of journals that are requesting more and more attention to sex and gender, especially sex disaggregated data and publishing, and even Nature journals made some kind of pledge some years ago. The Lancet has pledged to do this. There is a number of basic journals, even Cell Family has pledged to do this. Of course, it's imperfect. So if you come up with something that's super fancy and super new, and they think we'll give them a lot of PR, they're still going to publish it, even if you didn't consider sex and gender. Let's be honest about this. But in general, if you look at the pipeline, it is true that there is a growing attention to this, and they're asking their reviewers to look for this, which means that also when you're submitting papers, it might be more relevant and more of an issue you will be confronted with. So I think in general, there is a growing attention towards the subject, and I think researchers are feeling this, which might be something that you keep feedback to your universities. But as I said, and you know, money is usually, as sad as it is usually one of the biggest drivers to actually get them to do something.
Yeah, no, that's a really good point. And along those lines, we have another question from Yelena. She says, thank you for your presentation. How should we address the gender equality plans in our grant proposals? For example, how should we address the recruitment strategies, and how can we prove that we are not biased towards gender when we're employing? So you mentioned the Horizon Europe proposals. I think a lot of us are working on such of those, but also national proposals.
Well, there are a number of resources that the EU Commission has actually put online, where I can send you a link afterwards about those, on how to specifically address these aspects and what they're kind of looking for, and how to design these gender equality plans. There's a number of projects that have their own websites, so EU funded projects on gender equality plans, but there's also a number of central resources on how these things should be set up, and I think that could be most helpful in how to do it.
Yeah, we in our office at BNN, we're about 10 women and 2 men. So, you know, when we fill out our gender equality things, you know, we have to think about that as well. The next question is from Chloe. She writes, do you think that it might be too reductive to think of biological differences as driven by sex versus behavior differences driven by gender? It might lead to essentialism or under consideration of complex social processes on biological outcomes. That's a big question.
Yeah. No, it's a good question. It's a good point. I mean, I think you have to consider the whole thing as a process. We started out thinking sex and gender don't matter. Why should we look at them at all? And we will not overnight move into an arena where social processes, especially in the biomedical research field, will be given the attention they deserve to a certain degree. So I think it is where we are right now, I think, is we are in a moment where we need clarity about what it is that we're doing, because for a while we had conflation of what sex and gender is, and then what they're doing, and people really didn't have much of a clear idea of what they were doing. So I think right now the point is, first of all, create awareness about what they are, what they do, how you're measuring them, and what the limitations are in measuring them. I mean, it's as I said initially, it's not as easy. It's not like you say, okay, I take my female and male animals and compare them. The definition itself that you're using for sex and gender have an impact on the work you're doing. So I think right now we're at this phase where we really, at least I'm trying to plead for operational clarity of what it is people are doing. We need a lot more methodological attention in the work that's being produced. And then the next step is in trying to understand better how these processes are intertwined. I mean, you can do both, but the risk was that while people were kind of doing both before, it just meant lumping everything together. So I think right now we have to kind of map out what it is that we're doing, what it is that we're measuring, what can do what and what cannot do these things. And then from there, take a next step and trying to disentangle where we're seeing the impact of what on which other factors. And I think that's where we end up. And of course, there is, as I said in the slide where I showed Lori Heiss's review, there is an entanglement between both, and it is also difficult to completely take apart. But the risk is also that, well, by shunning the complexity, that we don't address it at all, with the consequences that we've seen in the medical field and we're still seeing. So I think right now we're really in a phase where we should focus on methodological clarity, on defining methodologies, on defining appropriate tools to actually do all of this. And then in the following phase, I mean, some people will start earlier, where some are already working on this and others will pick this up later. We can move to the next step and investigate all of the interaction with the social and biological determinants in more clarity. But these are also questions that have accompanied us forever. I mean, the nature-nurture debate, if you want, which is a simplistic version of this, has accompanied us forever. So it's just in a different, I would say, in a different realm that we're discussing this again with different methods.
Mm-hmm. Yeah, thank you. And for the attendees, feel free to keep writing in the chat if you want more clarification for some of these questions. This question is probably like asking a parent what's their favorite child, but is there are so many different factors at play in human health. Is there any area where you see there's the most dire need to be differentiating sex and gender data? We have partners that are working in creating medical devices, wearables, diagnostic devices, is of course people from the medical university. Is there any area where you think it's really the consequences are much worse at the moment, or kind of any hotspots at the moment?
Well, first of all, I think it matters everywhere. So that's the first thing I can say, that if you look at it, it's actually not something that is an add-on for something. It actually matters everywhere. What I think is an important point to keep in mind, we're seeing so much of a development in the digital and well, the digital space in general, and also in health technologies. And I think the risk is a bit of repeating what we've seen in medicine. So I kind of, the blindness I was mentioning and the bias that's baked into a lot of algorithms is something which I think is of concern, because we're developing all these nice new tools, and and then if you're a startup, you're under pressure, you have everybody's pulling you from all sides, you're bleeding money every day, you have to deal with these priorities, you have potential, well, hopefully future shareholders that you try to please, and so forth. So you're pulled in all directions. And the risk, I think, at this point is kind of re-ignoring the sex and gender component, as we've seen in I mean, we've done that in medicine. We showed how it's done, and we showed which kind of consequences it has. And this is kind of I see happening again in a lot of these new products that are being developed, that it's simply not considered. And I'm not talking about making your product small and pink. I'm really talking about the content part of how does this affect, for example, predictive algorithms. I showed you the chatbot, but there's many that go in the same direction. So how are you doing this? Which kind of data are you using? Are you actually considering any of this? Are you mitigating the biases that are inherent in the data you're using, and so forth? So I think the risk is a bit that we will see this all over again, but with more of a black box in between, because if you use some kind of neural network or machine learning algorithm that is self-learning, well, unless you have some kind of guiding, I would say, guiding and gating principles, you might just reproduce a lot of the things that we have, and that's, I think, where we could do better at the moment, especially with the fact that we're moving more and more in that direction.
Okay. And as you so nicely said in your guidelines for researchers, we have to spread the word, and this is what we're also interested in here. So you heard it. We have a lot of work to do. We need to get the information out there. So this webinar will also be recorded, and we hope that you can share it with any colleagues that might be interested. And we'll just hopefully be in touch about this topic in the next couple months. So if you're interested, you can follow us at BioNanoNet here. Join us at some upcoming events. These slides will also be available for our members afterwards. And we are hiring for a vacancy right now at BioNanoNet. You can check out all the information on our website and also subscribe to our quarterly. This comes out 4 times a year. Our digital magazine for developments in sustainable technology, where we'll also be reporting on this webinar. So thank you so much. It was really, really enjoyable from our end, and hopefully was beneficial to all our listeners. So thanks for joining us today, and we'll hope to be in touch soon.
Bye, thank you.
Bye-bye, everybody. Have a great day.